Hi everybody, my name is Elmarie Kotze. I'm from the University of the Free State. I'm a soil scientist. I mostly work on carbon and soil organic matter, and I'm going to try to show you a little bit of what goes on in my world. How does carbon work, and how is it related to farming? So this is the layout of my talk. I will look at the carbon cycle, and I will also tell you a little bit more about other cycles, like the nitrogen cycle, phosphorus cycle, for example. I will focus on photosynthesis and the role of the plant and the microbes, the symbiosis that exists between them. I want to focus a little bit on soil organic carbon versus soil organic matter. What's the difference? What's the similarities? What's the benefits that they will give you? I will then talk about how much carbon can be stored in South Africa specifically, and in the Western Cape as well, and then I will talk about measurements of carbon. So here is a diagram of the carbon cycle, and you'll see there's some arrows going in and out of this diagram. And as you follow the arrows, you follow the pathways of carbon. So carbon moves from the atmosphere in the form of CO2 as a gas. It goes to the earth via your plants in the form of plant photosynthesis that happens. And then that carbon is stored in vegetation in the soil before it returns back to the atmosphere by respiration. So there's two types of respiration that goes back into the atmosphere. It's either the plant respiration or animal respiration. And your animal respiration is then eating the plants, and then it respires the CO2 back into the atmosphere, while your plants are also respiring, and you also have root respiration taking place. So soil organic carbon is your main ingredient that stays in the soil. And then you also have anthropogenic activities on your right-hand side, you'll see on the slide, such as burning of fossil fuels, and there's also certain land use changes that have impacted upon your carbon cycle, and they've actually increased your atmospheric CO2 concentrations. Some agricultural activities have often reduced soil carbon concentrations. So let's focus on the role of plants in this process, and we look at photosynthesis specifically on your left-hand side of the slide, and the roles that roots play in releasing some carbon into the soil, as well as the respiration that happens via plants. So I'm going to focus on that now. So let's talk about photosynthesis and the role of the plants and roots in the carbon cycle. So for photosynthesis, you need energy that has to come in the form of sunlight. Then CO2 also is needed, and that is absorbed by the plants from the air. And then, of course, you need water, and this is absorbed by the plants through the soil. This then produces sugars in the form of glucose, which is then used by the plant to grow and produce crops and fruits. So as the plant absorbs the energy, the CO2 and the water, it produces this glucose sugars, and then this is what you'll find in the fruits. And they also need a lot of water. Plants are quite thirsty, so they all need those three ingredients to produce sugars. And in this process, they also produce oxygen that is then released by the plants back to the atmosphere. So now I want us to focus on the plant and the root zone and see how photosynthesis is involved with the carbon cycle specifically. So you'll see my little plant there with the leaves at the top and the roots at the bottom, and all those ingredients that I've just highlighted in the previous slide, where they go in and how they go out. So what is important to see is that the carbon, that the CO2 that comes from the atmosphere, the carbon fraction in that, goes into the plant, and then it becomes glucose, right? So then the plant uses it to grow leaves, and it grows the fruits and so forth. But about 50%, this is the most interesting part about the carbon coming in from the atmosphere to the plant, about 50% of carbon taken in by the plants in the form of CO2 is released by the roots in one or other form. This release of carbon by the plant roots drives all other nutrient cycles that you'll find, nitrogen, phosphorus, and any other nutrient cycle that you can think of. And there's a symbiotic relationship that exists between especially soil microbes and the plants, the roots of the plant, due to this release of the carbon. So it is quite interesting to know that about 50% of the carbon that is absorbed is actually then released by your roots. So now I'm going to show you the root tip. So this diagram over here is a root tip. And you'll see the root hairs growing off, and you'll see some things on this diagram, and I've numbered them from 1 to 6. So I want to focus on the root tip and the 50% carbon that was taken in by the plant and then released in different forms of carbon. For example, you'll see that part of the root tip and the cells can fall off, just like us humans lose skin cells. Those cells then of the root can go into the soil. That you'll find as a source of carbon. There's also soluble root extract number 3. Then there's volatile organic carbon. There's carbon in the form of mutualist, number 5, and when the root eventually dies off, then you get also carbon being released back into the soil. This is all sources of carbon that ends up back into the soil. Now, all these 6 carbon forms that you'll find being released by the root back into the soil, half of the carbon released is in, let's call it liquid carbon form. It's a sweet energy. It's also referred to as simple sugars, amino acids, organic acids, but it's a sweet, sweet energy. And this is extremely attractive to soil microbes, and it's almost like releasing kids into a candy store. This release of sweet energy by the roots leads to a symbiosis existing between the microbes in the soil and the plant, whereby both the plant and the microbe gains from this relationship. So let's imagine those red dots are bacteria. They are simple decomposers of these simple sugars that I just referred to, or this liquid carbon, as well as decomposers of nitrogen-containing compounds and organic acids. That's what bacteria does in soil. The release of the carbon by the root attracts the bacteria to the rhizosphere, and they love that sweet, simple carbon being released by the plant. So that's what bacteria does. They go for that sweet, easy obtaining carbon. So the next type of microbe that I want to show you in the root zone is fungi. They are also attracted by carbon being released by the root, and it's those green dots that you see on my slide. They are decomposers of more complex carbon chains of lignin, cellulose, or tannins, which the bacteria could not break down. So bacteria like sweet, easy stuff, and the fungi likes more difficult stuff to eat. So fungi feed on the more resistant type of carbon compared to bacteria. So the next type that you'll find is the hyphae. So fungi also form hyphae, which is root-like structures. So those are those purple lines on my slide. They extend into the soil, they collect water, and then they bring it back to the roots. So in exchange for the carbon from the plant roots, the fungi brings water closer to the roots. That's what the hyphae does. Then we also get rhizobium. So some rhizobium bacteria infect especially leguminous roots, and they form nodules where the bacteria then live. And in that nodule, they release nitrogen to the plant. So you'll see on my slide on the top of the root, I've made a little nodule, and you'll see the red dots is the bacteria living in it. So these bacteria then take organic nitrogen and they transform it into plant available nitrogen like ammonium and nitrate. And so this is where the nitrogen cycle is connected to the carbon cycle. But they would not have done this if they did not get that sweet carbon from the plant root itself. So it's again the symbiosis between the two. Top left, this is a photo of what a rhizobium bacteria nodule looks like. I'm pretty sure some of you have seen them. Then we also get another type of symbiotic relationship that exists between endo and ectomycorrhiza fungi that attach to the plant root. And you'll see that I've made those orange type of color, orange-brown, at the bottom right-hand tip of the root. They attach to the root and then they provide phosphorus and water to the plant in exchange for carbon. So again, what this mycorrhiza does is they'll take phosphorus, organic phosphorus, or they'll take fixed phosphorus in the soil and they'll convert it into a form that your plant can take up. So that is your phosphorus cycle connected to your carbon cycle. And again, it's all connected to the carbon cycle. There's a photo on the top left side of your slide that shows you what mycorrhiza actually looks like in the soil. Then there's another soil microbe called protozoa. It's indicated in those light blue dots. They are grazers of bacteria and they feed on those bacteria. In this process, protozoa then releases plant available nutrients into the soil, which is then available to the plant for uptake. So when the protozoa eats the bacteria, then it releases carbon and nitrogen and all sorts of other elements or nutrients that you can find in soil. Next, you will find nematodes. Nematodes are normally associated with plant diseases, but you will be surprised to know that about more, about 90% of nematodes found in soil is actually free living nematodes. They are quite positive, they have a positive influence, and what they do is they are predators, and they eat on bacteria and fungi, and also on some protozoa. So they eat all those bacteria and fungi, all those microbes that I've listed there. They feed on them. They also eat each other, and in this process of eating or feeding on all those bacteria and fungi and protozoa, they release plant available nutrients into the rhizosphere. So this is just a picture trying to show you how the release, imagine those yellow, green, and red dots that you see all over the slide. Those are nutrients being released in the process while you have all these microbes in your rhizosphere, and they run the whole show there. So all nutrients are locked up in carbon bonds and are released by the soil biota, decomposing these bonds held in organic or mineral form. So that's also what this picture wants to show you. The release of all other nutrients is driven by the carbon cycle, because those minerals are bonded, and it's attached to the carbon bonds are attached to these elements. And as the bacteria goes for the carbon, they release these nutrients, and it becomes plant available. So soil carbon is central in all of these cycles, as I've just mentioned, and the need for energy by all living organisms drives all other nutrient cycles. So carbon can be seen as a source of energy, and you also know it by what we're eating. If you eat carbohydrates gives you energy. So it's the same in the soil. Carbon gives energy, and this drives everything. All the nutrient cycles are driven by this. So let's move on to what is soil organic carbon, what is soil organic matter. Let's see if we can see if there's a difference between them. Soil organic carbon refers only to the carbon component of organic compounds. So that's why it's called soil organic carbon. It's only carbon. Soil carbon is a measurable component of soil organic matter. Soil organic matter is actually quite difficult to measure. And so labs tend to measure and report only soil organic carbon. They don't report on soil organic matter. So organic carbon makes up about 2 to 10% of your soil mass, and it has a quite important role in the physical, chemical, and biological functioning of your agricultural soils. Whereas soil organic matter contributes to your nutrient retention and turnover. It contributes to soil structure, moisture retention, and availability. It degrades pollutants in your soil, and it's quite important in carbon sequestration, and then of course soil resilience and soil health. So these two terms, soil organic carbon and soil organic matter, it's definitely not the same thing, but sometimes they are referred to as the same thing. So just remember that soil organic carbon is only a part of soil organic matter. It is not the whole picture. Now this is a diagram showing us the different fractions or components of soil carbon, and why I want to show you this is so that you understand when we get to the measurement of carbon why this is so important. So you'll notice that there are two main groups, green and blue. So right at the top, your orange is the soil carbon pool. That's normally what we measure in a lab if you want to measure total carbon, but it can be divided into the greens and then the blues. And the greens are your inorganic carbon. It refers to your carbonates, your limes that you normally found naturally in soil. You can get lime that was applied by hand, but mostly these carbonates are referred to as limes. So you can determine it, or you can see if you've got carbonates in your soil by using acid, a 10% hydrochloric acid test, and then you put a few drops on your soil, and if it effervesces, then you will see that you've got lime in your soil. Your pH will probably be above 8, 7.8 or above. That's when you get lime in your soil. On the left-hand side is your organic carbon, and this is the part that plays the biggest role in giving a certain function to soil and influencing your soil health and your soil quality. These components that you see here of carbon are mostly measured in labs and varies in the ease in which measurement takes place, and of course the cost involved. Mostly farmers and even researchers do not necessarily understand these different fractions and what they mean and how to measure them. So I'm going to try to shed some light on it, but it's important that you see that there's quite a few fractions of the organic carbon, the blue ones, there's quite a few fractions, and they all mean something different. So if you do total carbon, which is the orange, you have to be careful to make assumptions from that, and that is why carbon can be quite confusing. Yeah, I want to show you the components of soil organic matter. What do they look like? So soil organic matter makes up about 1 to 6% of your total soil mass. And your soil organic matter is composed mainly of carbon, hydrogen, oxygen, and it also has some small amounts of other elements such as nitrogen, phosphorus, sulfur, potassium, calcium, magnesium, and they are all contained in organic residues. Now, we can roughly divide your soil organic matter into 4 fractions, those 4 fractions that you see in the circle. You will see the green is the fresh residues. They are smaller than 2 millimeter. Fine roots, leaf stems, that's what they are composed of. Then you also get your decomposing organic material, your stable organic material, which is your humus, and you get your living organisms. So your living organisms is that blue section on your top right-hand side. It's about 10%. That's your microbes, bacteria, fungi. The living and the dead components can range from very recent inputs such as your stubble, or it can be decayed materials that are thousands of years old. Now, about 10% of your below-ground organic matter, such as roots, fauna, and microorganisms, they are living. That's about 10% of it. Right, so you'll see the humus part is the orange part. There's about 65%. That's what you would like in soil. And then you've got your resistant organic material, which is about 15%. So what is soil organic matter? These fractions of soil organic matter we can describe it according to size, turnover time, and also composition. So first on the left, you'll find dissolved organic matter, or DOM. And then you'll get POM, or particulate organic matter, you'll get humus, and you'll get resistant organic matter, or ROC. These 4 fractions differ in terms of turnover time, and there's vastly different rates in how fast they turn over. So you'll see, for example, DOM on the left only turning over within days, while your resistant organic carbon on your right-hand side in the orange square takes hundreds of years to turn over. They also make up different amounts of your total soil organic matter, with the humus organic material making up the most part, about 50%. Their composition also differs with the more fresher plant parts belonging to your 2 left fractions. So you'll see at the top, I've tried to explain to you what are they composed of and how much do you find of them. So this is the 4 fractions that you find. So these fractions that I've just explained to you, they are also cycled continuously within the soil, and soil organic matter can be in various stages in this cycling process. So the 4 stages can be described in this figure, with number 1 being inputs. So that's your plants and animals become part of the soil organic matter as they die or they create byproducts. Second, you get the transformation stage, where your soil organisms break up and consume your organic matter, creating different forms of organic residues. For example, you get fresh plant residues that are broken into smaller pieces that's smaller than 2 millimeters, and then it becomes part of your particulate organic matter fraction, right? So this material decomposes further, and in a smaller amount of more biological stable material enters your humus pool. So third, at the bottom, you'll get your nutrient release, where your nutrients and other compounds not required by microorganisms are released and are then available to your plants. So first, your organisms use the nutrients, and the rest are then released into the plants. Lastly, we get your stabilizing organic material. That's number 4. As the organic residues decompose, they become more resistant to further change. Now the question comes to mind, how much soil organic carbon can actually stay in the soil? So we have 3 things influencing this: soil type, climate, and management. This influences your organic matter inputs to the soil and its turnover or decomposition. So that's going to influence how much carbon is going to stay in the soil. So at the top, you'll find the potential storage of organic carbon in soil, which depends on your soil type. The soil type is basically your clay particles and aggregates that can reduce losses of organic carbon by physically protecting it from decomposition. The particles of organic matter can become absorbed to your clay surfaces, and which is coated with clay particles. All of these processes make it difficult for microorganisms to come in contact with your organic matter, and so therefore the amount of organic carbon stored in soil tends to increase with clay content. In contrast, if you have a sandy soil, microorganisms are able to much more easily access organic carbon and then cause great losses of organic carbon by decomposition. The potential storage of organic carbon, the top one, is soil, is rarely achieved because climate actually also reduces your inputs of organic carbon to soil. Then if you look at the attainable organic carbon, we're going to look at climate. Your climate determines the attainable organic carbon in soil by regulating plant production, right? So rainfall temperature influences your plant production. For example, under dryland agriculture, rainfall is the climate factor that has most influence on plant productivity and therefore inputs of organic carbon to soil. In regions of high rainfall, soil tends to have greater attainable storage of organic carbon than the same soil type in a lower rainfall region. So the more rain you have, the more carbon you can store. The temperature also plays a role. So with every 10 degrees Celsius increase in temperature that you have in an area, the rate of soil organic matter decomposition doubles. So moist, warm conditions results in most rapid decomposition of organic material. Now, although it is not possible to increase your attainable storage of organic carbon in soil, management practices actually also determines whether or not that attainable storage of organic carbon in soil is achieved. So these three things really play a role. So let's look at the management part. This is your actual carbon, right? So the management practices determines your actual storage of organic carbon in soil. How does this work? It increases your inputs and it can decrease your losses. So certain practices that you have can increase the amount of total organic carbon in your soil. So if you have increased plant growth, you will generally have increased organic carbon in your soil via shoot material, roots, root exudates, by giving optimal nutrition, fertilizer, increase your water use efficiency, decrease your diseases. Growing plants for longer periods each year, that generally increases your inputs of organic carbon. So if you have a shorter fallow period, conversion from cropping to pasture, conversion from annual to perennial pasture, this also all improves your inputs of organic carbon. Improving your soil structure, for example, can increase your amount of organic carbon stored in your soil because you reduce the losses of organic carbon from soil by decomposition and erosion. Because if you retain stubble, for example, you maintain ground cover and you reduce compaction by vehicles and stock. This all are management practices that increases your organic carbon storage in soil. So the question is how much soil organic carbon that enters the soil will stay there. It all depends on the soil type, the climate, and the management. And a combination of these three will lead to having more. So it means that you have to understand what soil type do I have? What is my bulk density? How deep is my soil? What is my climate? And climate is now changing. So you need to understand how to adapt to that. And you must adapt your management practice. And if you combine these three things and understand it, you will be able to keep carbon in your soil. So what is the impact that soil organic matter has on soil properties? So it is well known that soil organic matter improves your chemical, biological, and physical properties, as shown in this slide. This should be quite familiar to all of you. So you will see that there's a chemical, biological, and physical circles that I've drawn up here, which roughly quickly explains to you what does soil organic matter do in soil. It buffers the pH, for example, it complexes cations, it immobilizes pollutants, it binds heavy metals, so it bonds everything, it captures it. On the biological side, it's a huge energy source for biological processes, as I've explained before. It's a carbon source. So it's also a storage of nutrients, and it improves your soil resilience tremendously. And on a physical side, the structure is really, really improved, so you get stability in your soil. The water retention is improved. Mulching reduces your water and soil losses. It buffers your soil temperatures. So it's well known that soil organic matter improves these properties. Soil organic matter facilitates the improvement of soil by aggregating. This requires a regular carbon supply, of course. So retention of residual plant matter, cover crops, mulching, or by working organic matter into the soil. These are all ways that you can improve the soil. While soil carbon is really not essential for plant growth, it is beneficial for feeding. It's an energy source for your microbes, and it improves your soil moisture properties. and therefore, through plant available water, plant productivity, you can increase soil carbon concentrations by promoting sustainable agricultural practices. This also can, of course, sequester CO2 and reduce your greenhouse gas emissions. So this is just in brief, what is the advantages of soil organic matter. The biggest advantage actually of soil organic matter in a country like South Africa, where we have climate change and which has led to warmer summers and less and more erratic rainfall incidences that we've seen lately. So it improves the soil organic matter. It helps with your water holding capacity. This is because the addition of organic material increases your micropores and your macropores in the soil, so and actually the structure. And because it glues, it's like a glue, so it glues those particles together and it creates a very favorable living condition for my soil organisms. So in certain types of soil, organic matter can retain up to 20 times its weight in water. So because organic material particles have a charged surface, and this attracts the water. So it adheres to the surface like static cling, so it clings to it. And as a consequence, less irrigation water is then needed to irrigate your same crop. So for example, a 1% soil organic matter increase gives you about 25 millimeters of water in soil. So this equates roughly to about 250,000 liters per hectare more available water. And so 250,000 liters is about 50 big JoJo tanks. This is just by increasing your soil organic matter content with 1%. That really is one of the biggest advantages that I can see of soil organic matter in a country like South Africa. So let's see how much soil organic carbon is in South African soils. So if you look at this situation in South Africa, there's how much soil organic carbon is in our soils. This is a soil organic carbon content distribution map for South Africa for the top soil horizon. It's expressed in percentage by mass. It was done by Stronkost and Venter in 2008. And most South African soils are quite low in soil organic carbon, as you can see there. So low rainfall, warm conditions for much of the year, and sandy soils on top of that limits the buildup of stable soil organic carbon. So about 60% of organic matter in the top 30 centimeters of soil is located only in the top 10 centimeters. So that's why this map is only from the top horizons. So you'll see that I've said on the slide 58% soils in South Africa contain less than 0.5%. So that's quite low. 38% contain 0.5 to 2%, and there's only a 4% soils in South Africa that contain more than 2% carbon, and yet there you'll find them in the more rainy areas. So if you look at that map, it's also very related to climate, to rainfall. This is a table made by Swanepoel et al. In a review about the carbon status of South African soils, and it shows the different rainfall regions and how carbon is affected by the rainfall. So the Western Cape, I've tried to make a little cloud there. It receives about 850 millimeters a year, and so you can see that the carbon is quite low for these types of soils. So you'll see where the cloud is if you go down for the depths. So there's an average of about 1% of carbon in your 0 to 50 millimeters. If you go to 0 to 100, so that's 10 centimeters, there's just under 2% carbon. And as you go deeper down, of course, it gets lower and lower. So the Western Cape receives, as I said, about 850 millimeters per year, and you can see that the carbon is quite low. This is coupled with your sandy, stony soils that you find, so this is not very conducive for soil organic carbon accumulation. So if you think back to my few slides back where I said your soil and your climate and then your management, you can't do much about the soil that you have, you can't change it, and you cannot really do anything about the weather, but you can do something about the management, and the management is therefore important to then play with that and see if you can improve your carbons. So let's take a look at analyzing carbon in South Africa. Here is a diagram which I showed you previously of the different fractions, and I tried here to indicate how we measure it. So some of you might be familiar with some of these techniques. So what is important to see, as I tried to mention earlier, is that mostly total carbon is measured, the orange one. It's done by LICO, it's combustion, so you measure the total carbon. So the first question that you have to ask is, did they get rid of the carbon, the inorganic carbon? Did they make sure that there's no lime when they measured carbon? If you made sure about that, then you would know that your total carbon that you obtain by LICO is equal to your organic carbon, and then you can go further with that. So organic carbon, on the blue ones, they are measured with wet chemistry mostly, using Walkley-Black procedures, and some labs use the Mebius procedure. Once again, you will not have an indication of if this is stable or active or decomposed by looking at these values. So you only have the organic fraction. You don't know what the other fractions are. Furthermore, it's also important to know that most lab procedures are at most 65 to 70% successful in extracting all carbon within a specific procedure. So you only get an indication. You do not get an absolute value of what is in your soil. So that's important that you understand that. So total carbon is also relatively stable, and it's not a very sensitive indicator to use if you want to see changes due to management strategies applied. So in my view, the labor or active carbon fraction is much more sensitive to changes that takes place in the soil, and that's not that commonly measured. It's also it's not common for South African labs to determine soil organic matter, although it's a very simple procedure called LOI, or loss on ignition. The sample is simply heated at 500 degrees Celsius, and then weight loss is determined. And it's important to know that all analyses do not determine 100% of the carbon content. So on this slide, you'll see that the permanganate oxidizable carbon on the left bottom, that's your active carbon that you measure, and the arrow is very indistinctive, just randomly shows. It's not a specific block, but it's the active carbon. So it's the unprotected, but it's also some of it's protected, right? So you can get it out of there. What I also want to show you is the live organic carbon, the faunas and the microbial carbons. You can do it by PLFA, FDA, counting, respiration. Most of these analyses are really expensive, and so that makes it really difficult for a farmer to use and to try to get something usable out of it. I know that some of you have used the Haney method, and they've also used the Solvita method, where you use respiration. What is important in this is that you know that if you measure the live organisms, when you measure it, it's only a snapshot of what's happening in the soil at that moment. You cannot predict what's going on. In a month's time, it can be totally different, where your LICO carbon or your total carbon is more a long-time value. So all of these things you have to take into consideration when you work with carbon and you analyze it. I want to talk about estimating soil organic matter stocks from soil organic carbon. So because soil organic matter is not commonly determined in labs, it's usually estimated from your soil organic carbon analysis. So here is the way to do this calculation. It relies on the assumption that soil organic matter contains about 58% carbon, right? So this is debatable, but that's what research has shown in the early 60s, 1960s. So you take soil organic matter equals soil organic carbon times 1.72. Where does 1.72 come from? It's derived from 100 divided by 58, right? So this conversion factor varies in different soils. It can vary from 1.4 to 2.5, but the median or reasonable estimate is about 1.72 or 1.724. That's the exact number actually. And so that's mostly used 1.7, but you have to remember it can actually be 1.4, it can go up to 2.45. So the use of a single factor for converting soil organic carbon to soil organic matter is problematic, and it's not very accurate. So this conversion factor can be a problem. So some literature has proposed to use a factor of 2 rather, because this is based on the assumption that organic matter is about 50% carbon. It would be, in most cases, it'd be more accurate than the conventional factor of 1.724, right? So convenience, authority, and tradition, rather than strength of evidence, are in large part responsible for this widespread acceptance of this conversion factor. But I want to tell you that you have to be careful when you use the conversion factor, or you can use it, it's no problem. But remember that for example, in the Cape, Western Cape area, your factor will be much lower. It will be rather 1.4 than it would be 1.7. And if you go to a very humid area, then it will go up to 2.5. And so that's why they say a more accurate version is actually to use 2, right? So the other thing that I also want to talk about is converting your percentage to weight for a given depth and area, and they talk about stocks. So instead of expressing your value or your soil organic carbon as percentage, you express it as stock. So that therefore you need bulk density. If you have bulk density, then you can then work with that. But you need the depth of the soil where the sample was taken, and you need the bulk density of that specific depth. If you multiply it out, and there is ways to do it, so then you can work out how much is it in stock. So now I want to end this with the slide on how to measure and interpret soil organic carbon results. First of all, changes in your stable soil organic carbon generally occur very slowly. It's normally over decades, and it's often very hard to measure small changes against your relatively large background of soil carbon. Your changes in your soil organic carbon are largely determined by how much biomass is grown and retained above and below ground. So what you are growing, when you are growing it, that determines the changes. So a typical South African grain production system, where you will, let's say, yield 2 tons per hectare of wheat, you most often retain about 0.1 to 0.5 tons of organic material per hectare in the soil each year. And this again depends on what type of soil you have, if it's sandy soil or clay soil, but let's say it's 0.1 to 0.5 tons of organic material per hectare. This equates to a change in soil organic carbon in many instances of less than 1% of the total stock. So a larger change in your total organic carbon stock, which might take several years or longer to occur, is required before a significant change could be measured with any degree of confidence. Furthermore, changes in soil organic carbon are most likely to be observed only in the top 0 to 10 centimeters of the soil. So since soil organic carbon is highly dynamic, we need to find innovative ways to sequester more carbon by adapting strategies to our local conditions. So we need to sequester more carbon because it really holds, you can really save a lot of water, first of all. That's the main thing, if you can only think of that. So by monitoring soil organic carbon over time, the changes caused by management can be estimated. However, the fresh residues can vary widely. So depending on the crop or the pasture grown each year, when you are growing it, or you have cover crops, this will vary, and accurate measurements of changes in soil organic carbon requires a few things. You need to have a soil sampling strategy that captures the natural variation in your soil carbon, and then of course an estimate of your bulk density of the soil so that you can adjust for changes in soil mass at your specified depth intervals. That's where the stock measurements came into. So this then brings me to the end of my presentation, and I hope that you have learned something about soil carbon. Bye-bye.